with Make Community, and welcome to Plan C Live. This is an episode on RadVac, radvac.org, a radical vaccine for COVID-19, an open source vaccine. So a really fascinating development that we're really excited to share with you. I want to welcome actually Dorothy Jones-Davis from Nation of Makers, my co-host. And I'm also joined this week by Mike Sinisi, executive editor of Make Magazine, who just got that issue out and should be out to people that are looking for it. I want to welcome from RadVac, Alex Hoekstra and Ranjan Ahuja. Both of them will be talking, sharing with us about this development, which they're scientists, and there's a group of folks working on this, and we'll get to learn more. Before we do that, Dorothy, you had a little bit of information to share.
Yeah, so I wanted to follow up. Some of you guys have been following that we've been doing collaborative work with Get Us PPE, and we have some new grants coming down the pike. So if those of you that are interested in gown production, I know we talked about that now probably a couple months ago, I guess already. We are funding, so through Get Us PPE, we are funding a gown production cohort. And if that sounds like something that you might be interested in, next Wednesday, the 19th at 7 p.m., we'll have an informational webinar. I will stick the link in the chat here on Zoom, and we'll make sure that everyone that it's on YouTube also gets the link. And at the initial webinar, we'll just introduce an overview of what the program is. It's actually a cohort, so we'll be giving not just money, but also support. We'll also give an overview of what it takes to be a gown production facility and all the considerations that you have to think about, including materials, equipment, labor, and the like. And then we'll invite people to fill out an application to actually join if they're interested. And I also will stick that application link in the chat as well. But we do encourage you to apply if you're interested in even, or considering even becoming a gown producer, because gowns are one of the most needed pieces of PPE that we still have in our communities, and we really would love to see that need filled. So if you're interested in helping out and making sure that it's financially viable for you to do so, please do consider attending the webinar. I'll be sticking that in a second.
Yeah, great. Thank you, Dorothy. Well, let's get right into this, and I guess the formal name is the Rapid Development Vaccine Collaborative. So we'll get that. Alex, tell us what this is, how it came together, and why it's so important. At least we believe it's important.
Yeah, absolutely. Well, first of all, thank you for having us. It's clear that the Make community is unique insofar as its engagement with problems big and small, and COVID is clearly something that is inescapable across the world. So the fact that there are makers of every kind on every level engaged in this, the way they are, is amazing. I just want to thank the Make community for doing everything they can. And really, RadVac is our attempt to contribute to that community and contribute to a community of science in a way that is truly accessible, or what we hope is more accessible than traditional vaccine R&D, which tends to be a very proprietary, very guarded, and very sort of a high bar to entry in order to participate with. We believe that there are tens of thousands, if not hundreds of thousands, of very capable scientists around the world who have not just the skills, but the desire to engage with vaccine R&D. Our ambition is to unite those people with the resources of knowledge and an immense amount of background research into our design principles that they maybe wouldn't have had access to previously. So RadVac kind of began with an email that Dr. Preston Estep sent out to a couple of us back in early March, when it became clear, or rather it was becoming clear that COVID was going to become kind of an inescapable facet of life, not just for America, but in a truly global way. And really the email was about looking for projects that were pursuing fundamental solutions, fundamental biological solutions, namely vaccines, that were maybe already underway in a rapid development protocol, right? Looking for other projects like ours. And it became quickly apparent to us that to those of us who responded and wanted to dig into this work, that we couldn't find any quite like what we had in mind, right? There are a number of biohackers across the planet and people who are kind of DIY biologists, but there didn't seem to be a well-coordinated effort to really hone in on developing fundamental biological solutions to immunity. And so we decided to pursue this ourselves. So that's kind of how the RadVac team came together.
So just an email out to, you probably had some academic relationships and other things.
Yeah.
In the past.
Many of us got to know each other through work at the Harvard Personal Genome Project. Others of us have started companies or nonprofit foundations together in the past. So, we're a well philosophically aligned group. A group who, in the past has endeavored, both individually and as various groups, to build public resources for science and for scientists.
Yeah. What was the rough size of the group that originally got this going?
Oh boy. I think probably, maybe 4 people responded to that email chain. Yeah. And that 4 has kind of represented our core team. Obviously we've grown in terms of the number of collaborators who've come in, not just to help enhance the research, on a molecular biology level, but to engage with the research in their own kind of independent way, which involves making and taking the vaccine and using themselves as a test subject for it, which I think at this point, close to 30 that I'm aware of have done that, including me. I think I
tested it on yourself in the spirit of amateur science.
Indeed. Yeah, indeed. And I do want to be clear that, I'm a molecular biologist. I have a background in genomics and molecular genetics. I spent many years researching the molecular pathology of cancer and diabetes, and really trying to design proteins that could interrupt those pathologies by interrupting, signaling pathways within those diseases. So, it is an unprecedented project. This, RadVac is an unprecedented initiative insofar as its transparency and its inclusivity and its accessibility to, scientists around the world. But it is really founded on substantial, an immense pile of research that we've drawn on the academic and commercial expertise of hundreds of scientists, including ourselves.
You're actually sitting on that pile right now, aren't you?
I think I am. If I were, I'd be much taller. But,
just the whole floor is covered. Yeah, anywhere you go in the house.
Yeah. So, we do want this work to be accessible to both biologists and engineers on sort of every level, but the foundation of it is anything but amateur. One of the questions that we've gotten a lot is, or rather an assumption that people make about us, is that we're anti biopharmaceutical companies or anti-commercial vaccine initiatives or anti-FDA or anti-regulatory agencies or anything like that. We're not. In truth, those people are our brothers and sisters. We come from the academic background. We've worked with regulators numerous times in the past in various capacities. We've started biotech companies. We just understand that in order to develop the best vaccine possible right now, we think that it needs an approach that didn't exist in the conventional research and development pipeline. And a lot of that has to do with the ability to iterate and really develop the design in an iterative, progressive, evolutionary way based on an emergence of new research. So, for us, we began in late March, really. We met in late March and really began the design work based on what was understood at that point about SARS-CoV-2 and about COVID disease. And a lot of our attention was focused on antibodies. And at that time, much of the discussion about immunity and vaccines was focused on antibodies because antibodies are relatively easy to test for and are a critical component to immunity, right? A fairly well understood component of immunity. What has transpired since then is really an emergence of an understanding of the mechanics of T-cell immunity and really the targets of T-cell immunity in conjunction to B-cell or antibody immunity. But the importance of T-cell immunity has really come into the front of the discussion about a protective immunity that is both durable, that is something that could last for years, and cross-reactive if you are to encounter a second strain of the coronavirus. So I think it might be helpful to kind of take a step back into fundamental biology or fundamental vaccinology and kind of cover what is a vaccine, how do vaccines kind of, how do the various types work, and how does our vaccine in particular approach, the problem of or the challenge of creating immunity without introducing an actual pathogen. So,
yeah, I took myself off mute because I was thinking, okay, I come from a scientific background. I am following along. However, I'm not sure that everyone knows all of the differences and understands how antibodies work. That's an assumption that maybe we're making a little bit. So do you want to take a step back and explain a little bit? Let's start with some basic sort of biology about how does the body respond when we have a pathogen like SARS-CoV-2?
Absolutely, yeah. So vaccines are really meant to train the body's immune system, the host immune system, to both recognize and attack an invading pathogen, right? So in this case, it's SARS-CoV-2. It's a virus. It's one of the, I think, 7 strains of coronavirus that can infect humans. It's certainly it's something that we've dealt with in the past. Coronaviruses are fairly common. Folks on the call might remember the SARS epidemic back in 2003, or the MERS epidemic some years after that. Those were both coronaviruses. There are also some much weaker coronaviruses that are sort of the root cause of the common cold or some of the common colds. So coronaviruses are a fairly common virus in the viral world. But a vaccine is really meant to be, a way to train your immune system to recognize a specific pathogen so that when it encounters the real thing, it knows how to deal with it without it actually having to infect a lot of your cells and mount a significant disease. So there's a couple types of vaccines, and there's actually more types in development now than probably ever before. But the 3 major categories of vaccines that have existed previously are live attenuated virus, killed virus, and then subunit vaccines. And it's a little odd to think of the word killed in the context of a virus, because a virus isn't technically alive. It's kind of right on the cusp. Viruses require host cellular machinery in order to replicate, so that they don't quite meet the criteria for being alive. But essentially, an attenuated virus, or a live attenuated virus, is a virus that has been modified to be less pathogenic, less infectious, less virulent, so that your body has kind of a weakened version of it to be able to identify and remember, and so that when it meets the real thing, it has the ability to attack it. The killed virus, or inactivated virus, approach to vaccines involves, taking out the infectious or virulent genetic material inside of a virus. So just so that you have kind of the inactivated protein shell that makes up the rest of the virus. And in so doing, that protein shell is what's called antigenic. It elicits an immune reaction from your body. Your body can begin to identify various parts of that viral shell or that viral structure, and remember it, both in B cells and in T cells. And then the subunit vaccine, which is really, the best characterization of our vaccine approach, is various discrete elements of that viral structure, right? So the coronavirus, for instance, has a handful of proteins that make up different parts of that viral structure. The one that gets talked about the most is probably the spike protein. And the reason the coronavirus is called the coronavirus is because if you look at it under a very high-powered microscope, it looks like it has corona, which is made up of these little spike proteins that are on the surface. Those spike proteins interact with human ACE2 protein, which is a membrane protein on the surface of various human cells, like cardiac cells and lung cells and kidney cells. And that spike protein is really what binds to, like a lock and a key, binds to ACE2 protein and allows the virus to enter the human cell, or allows the viral genetic material to enter the human cell and become, pathological. So our approach is sort of a derivative of the subunit approach. We've identified, and I should say that dozens of researchers at this point have identified various subsections of these proteins, of these larger protein superstructures. Just the regions or the segments of those proteins that are specifically immunogenic, that are specifically targeted by the body's immune system. And we call these epitopes. It's just sort of an immunogenically active site on a protein. And what we've done is we've identified, or rather we've taken these identified structures and we've synthesized them, right? Just exclusively these structures, these little snippets of larger proteins. We've synthesized them, using a commercial service that synthesizes proteins, really small ones called peptides. They're usually, I think our average peptide length is like 21 amino acids in length. So they're very, very short, compared to the thousands of amino acids that make up these larger protein superstructures. Our peptides are very, very short. And for anyone with a background in protein biology or protein biochemistry here, you've probably heard that when it comes to proteins, form equals function, right? So the way that protein folds in its molecular context really matters. So we've had to do some design work to stabilize these epitopes, these peptide epitopes, into a structure similar to their native structure on the larger protein superstructures. But by doing so, we hope that what we've done is create very targeted antigens, very targeted immunoactive peptides that the body, that the host immune system can recognize if and when it meets them in the native structure, in SARS-CoV-2 whole live. So yeah, I hope that was a good enough explanation of sort of our approach to, designing immunity and eliciting immunity.
So when we hear about other vaccines, are they using those other approaches?
Some are. Many are using subunit approaches. Many seem to be targeting explicitly the spike protein, which again is a very prominent external immunogenic protein that presents itself to the host immune system very readily. But our approach includes not just the spike protein, but other proteins sort of elsewhere in the viral structure. And the reason we've done this is because, especially over the last 2 months or so, we've seen a lot of research on what epitopes, what active regions are eliciting a T cell response from the host. So the spike protein is really good at eliciting a B cell response, right? Producing antibodies. Antibodies help identify and recruit other immune cells to attack a virus or any kind of invasive, pathological enemy. But T cells are sort of the immune system's memory. There was a great paper, I think, mid or late July, that took cell cultures and samples from individuals who had previous exposure to SARS-1, so back in 2003, 17 years ago. And they subjected these patient samples to SARS-CoV-2 to see if there was, a response, if there was a recognition and an immunity, from one coronavirus strain to the next. And it turned out that the T cells that were formed during the 2003 SARS epidemic were responsive to SARS-CoV-2 and the COVID epidemic, or the COVID pandemic. So what we're seeing there is that there are regions that are not only conserved from other coronavirus strains, but they actually, elicited a T cell response that recognizes and provides an immunity to, or at least an immune response to this new coronavirus. So that gives us a really good insight as to how and where to engineer our approach to T cell immunity, which we know is a durable and cross-reactive response. B cells, antibodies in particular, don't typically last for more than a few months. I know in the press there's been a tremendous amount of attention paid to antibodies, and this growing concern that someone who is infected and recovers from coronavirus or from COVID might lose their antibodies over the course of 8 weeks or a couple months, and that's really because if the body is not being confronted by pathogens, there's no reason for it to continue producing antibodies. Antibodies are meant to be a very rapid response to clear a disease out. The T cells are really what form the sort of long-term memory of the immune system. So it's to be expected, in a sense, that antibodies don't stick around for very long, but that doesn't necessarily mean that one is susceptible to reinfection. If memory B cells and if memory T cells stick around and maintain that recognition, there's no reason to think that immunity is very fleeting. So many of the other, getting back to your question, many of the other vaccine approaches are exclusively focused on spike, and to me, I find this a little worrisome. Because spike, while it has a number of epitopes in there that are well characterized as eliciting antibodies and neutralizing antibodies in particular, spike is also the most mutable region of the coronavirus, which is it's the most frequently mutating, most high susceptible to mutation region of the coronavirus, which means if or when there is a mutation, it will likely be in the spike protein and likely in the receptor binding site of the spike protein. And that means that the antibodies that were specifically targeted at spike may no longer be useful or as useful, which can lend to something called, which is kind of a long-term safety measure or long-term safety concern called antibody-dependent enhancement. And this is where if you have non-neutralizing antibodies, antibodies that can still identify but not as efficiently eliminate or inactivate a virus, the virus can actually leverage those non-neutralizing antibodies to enter immune cells. Now, immune cells aren't typically susceptible to coronavirus infection. Immune cells don't express the ACE2 protein. But with the help of non-neutralizing antibodies, it's conceivable and risky that the coronavirus could leverage these, a future strain or a future infection could leverage these existing antibodies to enter immune cells and become much more pathogenic. So that, is something that concerns me. The other thing that concerns me about vaccines that really haven't adapted to, the ongoing research is that spike doesn't contain very many T cell epitopes. It contains many B cell epitopes, but very few T cell epitopes. Those T cell epitopes, the ones that we've characterized as being very high scoring in terms of their binding affinity, in terms of the number or the percentage of recovered patients that they appear in, they tend to be in structural proteins like the nucleocapsid protein, or in non-structural protein 7 or open reading frame 1AB, essentially not in the spike protein. So, our latest iterations have really honed in on these other non-spike areas that we think are really important, and the research is really building up as the evidence is increasing, showing that they're increasingly important.
So Eric Decker, one of our attendees, is asking exactly how does RadVac work, which I think you're explaining it, but I think sometimes
Sure.
Like you build a vaccine and you administer it nasally,
Sure.
a nasal spray, right? But what's in that vaccine?
Yeah. So our vaccine design that we've come up with has a grand total of 5 ingredients, 2 of which are salt and water. The other handful of ingredients are chitosan, which is a polysaccharide derived from chitin. Now, chitin is, I think, the second most common polysaccharide on Earth. It's found in things like mushrooms and in crustacean shells and shellfish and things like that. Chitosan, in the presence of a negatively charged solution, like our other ingredient, TPP, tripolyphosphate, forms these little nanoparticles, right? They self-assemble in the presence of this other solution, TPP. And what we can do is we can essentially have them self-assemble around our peptides. The 5th ingredient are the synthetic peptides that are meant to model, discrete pieces of the SARS-CoV-2 virus. So 5 ingredients: chitosan, tripolyphosphate, salt water, and our custom peptides. And I think at this point we're up to Ranjan, correct me if I think like 15 or so custom synthetic peptides, each modeled after a slightly different region of the coronavirus. And we're continuing to update that list, right? We're continuing to keep up with the research and sort of overlay, old research on new research and see, which epitopes are really well characterized as being immunogenic and as being sort of immunogenic in terms of non-neutralizing antibodies and in terms of memory T cells. And, we have a number of criteria that kind of lead us in the direction of, all right, this is a good epitope. So yeah, I mean, it is administered through a nasal spray, no injections. We chose that route for a number of reasons. One, because the mucosal immunity is tremendously important for a respiratory virus. Your nose and your mouth are kind of the first entry points of SARS-CoV-2 into your body, and so if the mucosal tissues immune system is primed, it's very likely that that's advantageous as opposed to a serological immunity, which is just sort of in your bloodstream. That once it gets past that point of infection, it makes its way into your bloodstream. You have a different part of your immune system that can then handle it. And the other part is accessibility. I don't think that we could get a distributed research and development done on this if people had to continuously inject themselves intramuscularly. It's just not a fun experience. Not that shooting a liquid up your nose is particularly fun, but it's certainly a lot more approachable than big needles or even small needles.
Yeah. So let's, again, you're a distributed group of people that you, you don't necessarily work in an organization, for the same organization. You're coming together to solve these problems. You came up with an idea of how to develop a vaccine different than the ones you thought were out there. You're not going into the business of producing that vaccine, at least what I understand. And what you've done literally is to open source the recipe for this vaccine with all the research behind it. Is that right?
That's correct. Yeah. I mean, I couldn't have said it better myself.
Okay.
We believe that right now, there's never been a greater need for collaboration kind of in place of competition.
Right.
Now, unfortunately, there is no precedent for a collaborative research and development of a vaccine.
Right.
The traditional authoritative approach is really to go through, a biopharmaceutical company and carry it through, multiple phases of clinical trials. And, we certainly hope that our work will make it into that level, whether, probably not by us, but pre that, right? Like it's pre-clinical trial, it's pre-authoritative pipeline. Again, we're not anti anything really. We're anti-COVID, and we're very pro accessible research. So, the idea here is really to make this a transparent process that is approachable and inclusive of a lot more minds. The thing that I think most of us or all of us worry about is that, humanity right now is placing its bets, or rather staking all of its hope on 200 or so commercial vaccine development projects, right? 200 or so commercial vaccines, most of which had to be developed and kind of locked in terms of their design back in March or April, before we really understood a lot of what we now do understand about the mechanics of immunity to SARS-CoV-2. So, oh, go ahead.
No, I was going to say that's exactly what I was thinking. Is like, the ability to sort of iterate is enhanced with the scenario that you set up, right? So you have all these great minds. They're not in one place, right? So you don't have to have the luck or the chance that this group of scientists would happen to be at the same group and working together. And certainly in these kinds of situations, we know that scientists all over the world are collaborating. One of the things that's really exciting is that journals have become open source by necessity, right? So everybody who had paywalls for all the articles are like, no, we need to open up the science so we can make sure that everyone can share. And I think that this takes it to another level. It's like, how do we get the best and brightest, no matter where they're at, thinking about this really, really horrible problem that we have and iterating in a way that's rapid enough that we can come up with candidates. And what I really like that you just said is that, the hope is not necessarily that, we're going to always be brewing this in our basement and people are going to be coming to your garage to get, your vaccines. It's that a pharmaceutical company might see the promise in one of these strategies, take it on as a candidate and push it through faster than they would have had they had go, searching through just their limited portion. And that's kind of what it sounds like, right?
Yeah, you nailed it. I mean, truly, it's safety by distribution, right? It's safety in numbers. We really hope that, the thousands of scientists and engineers out there who are capable of doing this work now have an avenue to contribute to it, right? Whereas before, if you weren't in one of these 200 or so vaccine companies, doing the R&D is not really accessible. So our hope is to provide a resource that catalyzes a distributed involvement in this.
Yeah, and I might add that the way the economic system is set up, it means that certain parts of the world have access to pharmaceutical companies to develop vaccines and other parts of the world don't.
And to receive, and this strategy allows people to also receive the vaccines that may not normally be the top of that
Right, exactly,
incentivize
and to develop it potentially. So,
it. 100%.
so Pablo Castellanos in our Zoom group asks a question, which leads us in a good direction here. How can other groups that are interested in small batch testing of your vaccine participate and add value to the RadVac project?
Yeah, Ranjan, you want to take this one?
Yeah, absolutely. I find the vaccine itself a beautiful metaphor for what you referred to earlier, Dale, which is that we don't have an organization, but we sort of self-assembled around this purpose, and you can do much the same. If you have a local group of people that who are interested in taking this on, self-assemble on it, and go through the white paper. Dig into the science. Pick up the science that you're not familiar with yet. And get in touch. Get in touch with us. We're trying to organize all the groups that are getting in touch with us so that everyone can freely communicate and exchange ideas globally.
Yeah. Now, you don't, I mean, this is not necessarily something that everybody can do themselves or should do. Yeah. But Alex, you were telling us, the equipment needed to produce this vaccine is not, it's not out there. It's pretty humble.
Yeah, absolutely. One thing that we've done recently is buy a $29 magnetic stir plate, which, if you're not familiar with it, is literally just it almost looks like a hot plate, but there's a little magnet inside that if you have a separate plastic coated magnet, it sits on top inside of a beaker and it spins, right? And it's meant as a mixing device. We bought a $29 one on Amazon or eBay or something, and we compared it to our $400, lab grade magnetic stir plate, and it works just as well, right? So the traditional barriers to doing
the physics doesn't change.
right, exactly. Like a magnet's a magnet, a stir plate's a stir plate. So to some extent, if you wanted to invest in a really nice set of pipettes that was really high precision and truly like BSL 2+ lab grade, you could. Or you could save a lot of money and buy a $100 pipette that is 90% as good. I would caution anyone who wants to do that to calibrate your tools, to test them first, to approach this with rigor.
Right. And I kind of want to make this point because DIY sometimes get used as like, well, you just go in your home lab and make it. And it's not that so much as that the tools are accessible to people who have the knowledge and experience to do it. And those could be, I think you said that there's thousands of labs around the world that could make this, right?
Absolutely. Yeah. We're really here, not necessarily to develop a DIY vaccine, right? Like the intention for this is not to live through all time as a DIY approach. Right. It is to lower the barriers for access, to include more brains in this shared problem. Yeah. And to distribute the R&D in as efficient a way as possible, so that it doesn't have to live in 1 or 200 institutions' heads.
Yeah. Right. Alex, that's one of the things actually I think is really interesting. Is part of what you're doing is disseminating information and research from other groups as well, and doing it through an open source platform. You guys published your research with a Creative Commons license, which I think is really fascinating and very novel in itself. But touching on something that Dorothy had said before, that a lot of the journals and the researchers are now putting their information out in the open source capacity, so that this information that is the foundation of how you are able to gather a lot of the cutting edge research and information that's led to the progress that you're at with this stage of the designs that you're doing. That's how I understand it, correct?
Absolutely, yeah. I mean, we have truly benefited from the open source atmosphere into which we entered. We are by no means the only open source scientists in the world, right? I wish there were more, but I'm immensely grateful for the journals that have lowered their paywalls, even just temporarily, for COVID research. We are in unprecedented times in terms of access to knowledge, right. And our hope is really to lower those barriers more.
Yeah. By the way, I don't think we made the point. You've written this up as a white paper. It's available through your website on radvac.org. This is what open sourcing means in this case, is to show your work and to back it up with research. We may be able to put that link directly in the chat here. But Alex, you said a lot of people, some makers and others are contacting you. Would love to hear a little bit about how that conversation is going and what you're thinking there.
Certainly, yeah. Ranjan has actually been leading a lot of those conversations.
Okay.
So Ranjan, I'd love for you to step in.
Yeah, thanks. Yeah, we initially got, so this project is relatively still not very well known. If you look at how many people there are on Earth compared to how many people we've reached, it's a tiny fraction. So we have gotten a number of different responses. We've gotten quite a number of responses from non-scientists, people who just want to know how, when this will be available or how they can get their hands on it, because there is this incredible sense of urgency. We all feel threatened when we go outside, and sometimes when we don't go outside, about our health and the health of our families. So there's been a lot of response to it, in that way, which is that people recognize, I think they intuitively recognize that this is something that needed to be done, and that it serves a purpose. Just waiting around for things to get better or for vaccines to appear in a year is innately dissatisfying. The other type of response we've gotten is from medical doctors and professional scientists who share the first set of concerns, and then think, well, I've got some skills. What can I do about it? How can I contribute? And some of them want to, offer suggestions on the vaccine design and leave it at that. Others want to actually replicate the setup. They want to start doing some experimentation themselves, and perhaps fork the design. And the open source structure enables and encourages this. Take it, make it your own, take it in a different direction. Put your ideas in there, try and improve it. According to what you think might work better. And some of those folks are what you'd call biohackers, who have already got experience in biohacking. Others are medical professionals who just want to take this on as an N of one sort of thing. So it's been a variety of inquiries, and it's really encouraging. This is exactly what was intended. It's the agility or nimbleness of this approach is not to be underestimated.
Yeah, that's great. Alex, I have to get slide this in, because in our previous conversation, you used the line, "A vaccine is a hack," which I think
that's true.
If we don't understand anything else, we don't understand that much. I think what you mean is you're going in almost behind, in the back door. You're breaking something and replacing it with code that will work to make things that work the way you want them to work, right?
Absolutely, yeah. I mean, if we're any vaccine is introducing an exogenous piece of code to reprogram the immune system to do something it wouldn't do otherwise.
Yeah.
Right. And it's a benevolent, productive, positive hack. And so I think in that sense, anyone who makes or takes a vaccine in any capacity is a biohacker.
Yeah.
So
another question is just how has your understanding of COVID-19 changed by all the research you're doing? I think for the lay person, it's hard. We can kind of see what it's doing, but we don't understand a lot about this virus. And
sure.
And I would guess even the scientific community is trying to, is in much the same place. They may understand the class of things, but this is a distinct thing, and it operates, it behaves in a unique way.
Absolutely, yeah. Like I said, there's 7 I think 7 or so known strains of coronavirus that can infect humans. This one is unique and really nasty in a lot of ways. The efficiency with which the ACE2, or rather the efficiency with which the spike protein's receptor binding site binds to human ACE2 cells or human ACE2 proteins is really efficient, right? That is one of the most distinct things about this virus. ACE2 is an incredibly common surface protein across human cells, right? So cardiac cells, liver cells, obviously lung and respiratory cells. Each one of these tissues is sort of uniquely susceptible to this virus. And what we're beginning to see in an unfortunately increasing percentage of the population are really long-term health effects, even in recovered patients. So, I watch the number of new cases and the number of reported recoveries every day. And I take that second number, that number of reported recoveries, with a big grain of salt, because we're not yet, clear on the true outcomes of this disease in a long-term way. I think in a similar way, we won't have a good understanding of the long-term safety of most of these vaccines for a little while too. The accelerated approval process, the accelerated trial process of these commercial vaccines is such that there's no real guarantee of long-term efficacy or long-term safety. So these are things that we really have to consider up front, and we have to design for these things up front as best we can. Much of our work thus far has been in reducing the possibility for long-term health effects and long-term enhancement effects by choosing very discrete design elements that address those up front. But we're in uncharted territory, and we're not very far into it. We've only just begun.
Yeah, and you've only had limited trials, but one person's asking any observed negative reactions to the vaccine, which could, I was determined that biologically, but it could be a media reaction, it could be other social reaction to the idea that this is out there now. Sure.
There's definitely been a strong reaction amongst the bioethical community, who take a strong opposition to any derivation or any deviation from the authoritative research track, right? And we are all scientists. We were bred and trained into that scientifically rigorous approach to adhere to those bioethical standards and to those standards of research rigor. We also know that sometimes it's unethical not to run the experiment, right? And we've done everything in our power to reduce the risk that we pose to ourselves, and eliminate the risk that we pose to others. I think one thing that we want to be very clear about to anyone who that we help take the vaccine, or any one of us who takes the vaccine, is we also, we don't change our behaviors necessarily to be more risky. We're still wearing masks, right? We're still washing our hands. We're still socially distancing. One reporter quoted me as saying that I wasn't going out licking doorknobs, which is true. I never did that, but I'm certainly not going to start now. We're not at a point of,
you're not invincible, in other words.
Right. I don't expect myself to be invincible. And I certainly don't want to modify my behavior in such a way as to put others at risk. So I think, a fair way to characterize our, go ahead. Sorry, Dorothy, did you have a question?
Oh, no, it's okay.
Oh, okay.
Yeah, I was just going to say, did you have
any?
I think I'm cutting
you dropped out there, Dorothy. Yeah.
I can hear you though. I think I can hear you.
Can you hear me now?
Mm-hmm.
Yeah.
Okay. No, my question was just going to ask, have you had any positive, like reactions? Are there unexpected connections that were made, or people that were very positive about what you've been doing, instead of focusing only on the negative?
Well, I would say that the response from the community of independent scientists and makers, and even amongst, I mean, our group is largely made of people that we've known through our academic and biotech connections over the past, collectively many decades. So the response from some scientists, both institutional and otherwise, has been very positive. And I hope that as people begin to wade into, the real sort of technical meat alternative of our white paper, and begin grappling with these technical principles, that will have a more rich conversation around, okay, can this work? Not just in one person, but can this work in a wide population? Can this scale to 1 billion doses? Can this ship or be made in low access or low resourced locations? These are all questions that we've yet to address. We've been very honed in on doing our homework and trying to design a very well designed vaccine. And really there's a tremendous opportunity for us to learn from a wider community. Whether it's the maker community or the biohacker community or academic and commercial vaccine developments, we're here to listen as much as we are to teach. I hope that, I hope that there are people out there who, have the ability to teach us.
Well, let me ask, that leads to a question in which we've discussed a little bit, but what we saw when the maker community was doing PPE is, yes, there were scientific and technical people doing designs, and there were other people figuring out how to do production. But there are a number of people that were really critical to the success of the programs that I just call organizers, for lack of a better things, but often they were the interface between those people and a broader community. They helped to spread knowledge that such PPE was available. They helped to figure out distribution networks. And so is there a role for people who wouldn't know a peptide from a peppermint, and know how to help you?
Yeah, I mean, absolutely, right? We, we have a very specific skill set in our core group, right? We are molecular biologists, and to some extent, we've dabbled in community building, and some of us are great at community building. But the hope is not to stop at building the community. The hope is to learn from that community and to gain from the inclusion of diverse ideas, or a diverse set of ideas. A community in and of itself, is not particularly valuable. It's what that community comes together to do, and what it can learn from other parts of itself.
Yeah.
So Ranjan has really been critical in heading up that community effort, and
so Ranjan, people could go to the website and find some ways to express interest.
Drop us a line. Yeah, there's a contact form and an email there. The connections are really crucial. Because this is growing organically, any given person out there who now knows about the project and who's interested in seeing it progress and improve, can contribute by thinking of the folks that they have around them, the people that they know, and who they could tell about it. If you know scientists who might be interested, or biohackers, or people who are technically adept, with these skills, introduce them to this, and maybe they can get involved, on the technical side. This is so we've gotten a lot of inquiries about, can I get the vaccine? Can I take it? Can I join your clinical trial? Again, this is totally outside of the typical structure. So we don't have a clinical trial where a lot of this work has been done from the literature. The testing has been done on ourselves, ends of one, self-experimentation, self-administration. And we actually don't that we're just sharing that that's what we've done, but we're not running a trial. So that is not, what we intend for people to generally do.
Yeah.
But the connecting is critical, because we realize that these ideas often come from outside the box. So if someone has a background in chemistry, they have a very different skill set and a very different set of experiences than someone who's purely a biologist. And they may be able to see things that the biologists may miss in the formulation of the vaccine, or in the delivery, or how this might interact with the immune system, at the cell surface. So, that is also something that we're after, is to get people involved who are not typically involved in vaccine development.
Yeah, absolutely. And I'll extend that by saying, I hope that there are people and institutions out there who will carry this through, the authoritative clinical trial processes, right? And we'll develop this in the authoritative track that where it's possible to scale to 1 billion doses. And I hope that they do that with accessibility in mind. I'm not even opposed to people, patenting, their own version of this work, and just leveraging the R&D that we've done so far. If someone makes $1 billion on this, I'm happy for them, right? Like, you can send me a check for a penny if you want, but our intention for this is really to enable the most amount of people in whatever way they want to be enabled.
Right. It's almost also to make sure the best science happens in the best way possible.
Absolutely.
And not necessarily, it doesn't have to align with market or political interest. Let me, Pablo Castellanos again asked a really good question. You might look at it in the Q&A, Alex, because I can't read all the details to it, but he's doing some research on producing small batches of the vaccine already. But he's wondering, thinking it could be very expensive to the minimal amounts of synthetic peptides available in the market. I just thought I'd see if you want to address that.
Yeah, absolutely. So, the synthetic peptides are probably the most, not necessarily cost prohibitive, but definitely the most costly element to our vaccine approach, right? Our design. There are numerous synthetic peptide providers, right? Commercial peptide producers, synthesizers out there. Obviously you want to be mindful of their quality and their purity. But what we've done is really band together as a collective of individuals to pool our resources, our funding, and our data. So that, by working as a team, by working as a group, not only have we lowered, kind of the cost barriers to this, but we've also, begun to pool our data in such a way that the data make more sense together and are more robust and scientifically rigorous together. If we all do this as an N of 1 study, we may not learn much. For us, the power really is in large numbers of participants. So we're working on growing our own cohort, our own sort of group of individuals that line from, I think it's Monty Python and the Holy Grail, where one of the peasants says, I thought we were an anarcho-syndicalist collective of, and I didn't vote for a king. That's kind of how I characterize our group. We are a collection of individuals. Yeah. And
do you think it'll ever form it into an organization of some kind?
Well, I mean, we formed a nonprofit, right? Just because, for us, it made sense to pool our financial resources and maybe to give people kind of a channel to support that work. Good. So RadVac is a 501c3 nonprofit, formed in Massachusetts and registered in Massachusetts. In terms of pivoting to a commercial vaccine developer and the kind of entity that could carry this through the clinical trials on our own, I don't I don't necessarily foresee that. I think, that process takes a tremendous amount of resources that at present we don't have,
Right.
nor do we have really the expertise to do that, right? I think there are other people out there who may not be vaccine developers, but maybe clinical trial organizers and clinical trial managers, and really have a good view of that infrastructure and that process.
Yeah. Well, we're up on the hour here, and I want to thank all of you. But I just really, for Alex and Ranjan, for sharing with us. But what you've done is really a precedent, I think. Unprecedented rather. And I hope, apart from just the vaccine, this model of distributed development, which we've seen throughout, through the maker movement and the PPE and other things. I think this is one of the things that I hope can carry on and be applied again and again by other people, saying, as you did, there's a lot of smart people out there and they're not necessarily collaborating. So how do we change that? And, we can work on on big problems that way.
Yeah,
you hit it on the head, Dale. Yeah,
absolutely agree. And I want to be clear. I don't think that, and I'll speak for myself. And I don't want to throw any of my other, fellow collaborators under the bus. But this really didn't take a special amount of genius, right? What we did was a tremendous amount of homework and resourcefulness. But we didn't, we tried to be as uncreative as possible in developing a vaccine. We tried to be as well evidenced and as evidence-based and empirical as we could be in following the data and the research to lead us in these design directions. So we will continue to do that, right? We will continue to follow the research and update based on the best information possible, and continue to share it so that other people have access to it in a concise way.
You're sure you're not part of a conspiracy?
I would tell you, but then it gets complicated.
Yeah, exactly.
We got rules.
Yeah. Well, good. Well, Alex and Ranjan, thank you very much. Mike and Dorothy, thank you for joining me. And,
thank you.
We really appreciate it. Good luck, and let us know if there's anything we can do to help, or if there's any breakthroughs that we can report on.
Yeah.
Thanks a lot.
Absolutely.
Take care.
Thank you all so much
Thank you all for joining.
Yeah. Take care.